Hello, my name is Andresa Berretta, and I am here today to share with you what we are doing using propolis in anti-aging, especially antioxidant effects that can protect against several important degenerative diseases. I would like to thank you to Dr. Bridget Goodwin for this invite. I appreciate it a lot. And it is a great pleasure to be here with several other great names of Apitherapy universe. Thank you very much. Congratulations for all the scientific committee and the organization for this Apitherapy Australia conference. Thank you. I will share my screen so in order to start my presentation. The title of my presentation is Antioxidant Effects of the Oral Use of Propolis Extract, a Before and After Clinical Trial. If you need, you can be in touch with my email contact that is presented here. I would like to start talking something about oxidative stress. Maintaining cell viability requires a continuous process of energy production, which is largely accomplished by mitochondria. In this process, reactive oxygen species are formed, many of which participate in the control of cellular homeostasis, acting as biochemical intracellular signaling or even controlling gene expression. Under conditions of hypoxia, aging, or cell damage, this respiratory chain may be decoupled, leading to the formation of reactive substances such as aldehydes, malonaldehydes, superoxides, and peroxides, which in turn damage nucleic acid as DNA and RNA, and cellular membrane. Under physiological conditions, there is a balance between free radical production and its elimination. However, excess production of oxygen-free radicals defined as oxidative stress can lead to cell damage and be part of a number of some chronic degenerative diseases, as observed in humans, such as cancer, inflammatory and cardiovascular diseases, diabetic mellitus, atherosclerosis, and Alzheimer's disease. Antioxidant defenses are composed of enzymatic mechanisms such as glutathione peroxidase, catalase, and superoxide dismutase, and dozens of substances that play a role in binding and neutralizing free radicals. These substances may be endogenous or offered from diet or supplements, as flavonoids, for example, carotenoids, polyphenols, ascorbic acid, and vitamin E. In complementary medicine, the use of natural antioxidants-rich substances is frequent, mostly polyphenols, and B products are inside of this group. I will now talk something about antioxidant in vitro studies, because in vitro studies, in the literature, several studies already was performed to evaluate in vitro antioxidant action of diverse propolis types and different propolis extracts. Here in this publication, done by the group of Dr. Yala and Suzanne Valim, entitled Effect of Brazilian Green Propolis on the Production of Reactive Oxygen Species by Stimulated Neutrophils, they evaluated propolis ethanolic extract antioxidant action and also some fractions in order to try to evaluate the most important compounds involved in antioxidant action. This study is little old, however, brings some important highlights. Here in this study, the performance of antioxidant action was done using chemioluminescence. And we have two different models that was stimulated. In this case, neutrophils was stimulated by zimozan in the presence of luminol or lucigenin. Here, the control used in this protocol was quercetin. Quercetin was presented the better antioxidant effect in this model, in both cases. And different extracts, or I can say fractions, because in this study, the ethanolic extract were prepared and fractions of this ethanolic extract was obtained as butanolic fraction, chloroformic fraction, and hexane fraction. And the ethanolic extract behavior is this worst one, and the fractions as butanolic and chloroformic was the best one. It was quite similar in both models, with some more divergence results in the low concentrations, but the conclusion of this work was that the fractions, the butanolic and chloroformic fractions, obtained the best antioxidant action. In order to try to elucidate the compounds involved in this action, they isolated camphoride, isosaccharinetine, aromodendrin methyl ether, and also the 3-prenyl paracumaric acid. And the results demonstrated that the camphoride was the best one, demonstrating quite equivalent effect of quercetin. Here I am presenting the isolated compounds that were studied: isosaccharinetine, camphoride, aromodendrin methyl ether, some flavonoids, and some phenolic acid derivatives as paracumaric acid, 3-prenyl paracumaric acid, and 3,5-prenyl paracumaric acid. This was the compounds evaluated in the fractions and in the extract. The butanolic and chloroformic fraction are presented here. In fact, I changed this. First one is the chloroformic fraction, and the second one, the butanolic fraction, demonstrating that paracumaric acid, aromodendrin methyl ether, here, 3-prenyl paracumaric acid, isosaccharinetine, camphoride, and 3,5-prenyl paracumaric acid, or more popularly, articulin C, is present in this chloroformic fraction, and in this butanolic fraction, we have some other compounds, and here we don't have the camphoride and the articulin C, and but the other compounds were responsible for antioxidant action too, as it was demonstrated in the last figures. So camphoride was the most impacting effect on antioxidant model in this model, and so butanolic and chloroformic action demonstrated the high inhibition of oxidation process and camphoride. This cytotoxicity assay was done in order to demonstrate if the cells would be killed, and because of it impacting the results. However, all the results or the measures done demonstrated that don't have the quantities used not killed the cells, so the effect observed was a consequence of the result, the antioxidant result. So I would like to introduce the antioxidant clinical trial that we did in partnership with Dr. Eduardo Barbosa Coelho from the Faculty of Medicine of Ribeirão Preto, University of São Paulo. We did this study using university structure and the financial support from CNPq, FINEP, and Apitherapy. We published this article in 2020, and I would like to share with you now the results. We used in this study the green propolis standardized extract EPPAF. In Brazil, we have several types of propolis, and we prepared a blend around 20 years ago after evaluating each kind of propolis that we have in Brazil under the chemical point of view, but also biological point of view. And after this, we prepared a blend that we used in all development and all production that we do. We used all the same extraction process after doing a deep study from polar solvents until apolar solvents, defining which range of hydrochloric solution is great for our purposes. And so we always perform hydroethanolic extraction from propolis raw material to have our ethanolic extract that demonstrated that we can demonstrate reproducibility batch to batch under this chemical profile using HPLC techniques and also Singh-Lawyer chromatography in order to demonstrate that some compounds are reproducible batch to batch in a range specific. Of course, we never will talk about identical propolis extract when we are talking about natural products, but we can have some reproducibility under some range. We approved this patent letter for this composition some time ago in Brazil. And in this study, we are using tablets using EPPAF dry extract. In this case, each tablet possesses around 125 milligrams of propolis dry matter. The recommendation for the first step of the study is 3 tablets, and the second stage is 6 tablets. So we have a 2-phase sequential study, open label, non-randomized, before and after clinical study. What it means that we are using the same group to be the control of this group after treatment. So we don't have 2 groups to compare or 3 groups. We have the same group of volunteers that is healthy to be controlled before and after treatment with 2 different dosages of propolis EPPAF. We used here healthy volunteers. So it is very important because our intention was to see if propolis is administered to healthy volunteers, can we protect from the oxidation damages? So the idea was to use or to suggest the use of propolis as a protective from oxidation process. So was healthy volunteers. So in the beginning of the study, blood and urine sample were collected from the volunteers, and so they started the treatment of propolis EPPAF during 7 days by oral route. After 7 days, another collection of blood and urine sample was done. And so the next stage started, that was with 700 milligrams of propolis EPPAF per day during, again, 7 days, where we collected the samples of blood and urine sample. And after 30 days of the stop of the process, another samples were collected in order to evaluate the safety. The inclusion criteria of this study was no history of allergies, no continuous use of medication 1 week prior to the study, read and sign the informed consent form. The exclusion criteria was pregnant women, women breastfeeding, smokers, and participants with history of alcohol and drug abuse. The outcomes of this study was the primary outcome, antioxidant efficacy that was measured using urinary excretion and quantified 8 isoprostanes, that is a lipid peroxidation biomarker, and 8-OH-GG, that is 8-hydroxydeoxyguanosine, a biomarker of DNA damage. Until we know, this was the first time that this biomarker was studied in a clinical trial using propolis as treatment. The secondary outcomes was the safety of EPPAF. For this, we evaluated the biochemical parameters of kidney and liver functions. And as exploratory outcomes, we evaluated the biochemical pathways involved in oxidative stress, as the determination of FRAP, vitamin E, superoxide dismutase, GSSG, GSH plasmatic, and malonaldehyde plasmatic 2. This study involved the recruitment of 56 patients, and in the end, only was allocated 37 patients. The mean age of these participants was 30 years old, healthy volunteers, and 67.7% were women. And some discontinued the treatment, 3 discontinued, and 4 because declined to participate, and some because of headache or diarrhea. And in the end, from 37 recruited, only 34 concluded the study. About the safety aspect, both the groups after and before the treatment possesses similar parameters, demonstrating that the 2 doses usage and the period of treatment not compromised the health or the safety of these patients, these volunteers. The use of propolis EPPAF was safe. When we look to the outcomes, to antioxidant outcomes, we can see that propolis exerted an effect in GSH improving this value with the treatments in the both cases, both dosages, and a superoxide dismutase also were improved, demonstrating that EPPAF can be acting by the improve of this enzymatic function in the two dosages studied. Here you can see the superoxide dismutase effect in two dosages, and it was possible to correlate superoxide dismutase enzyme with 8 isoprostanes effects, as can be seen in this graphic of correlation. When we evaluate the 8 isoprostanes, we can see that it was not dose dependent, because in the both dosages used, 375 milligrams of EPPAF and 750 milligrams of EPPAF, we have the same effect. So the lower dosage is enough to protect the patients of lipid peroxidation. In the case of 8-O8-DG, that is the biomarker of DNA damage, the most important effect was with the higher dose, that was 750 milligrams of EPPAF per day during 7 days. When we look here in the table, it's possible to see the great difference that we can observe. And in the case of 8 isoprostanes, the best effect or the enough effect was observed with the low dosage, and there was the equivalent in the higher dosage. And in the other case, in the protection of DNA, the higher dosage was best. This study possesses some limitations. Its design as a before and after study is a limitation because we are not controlling it. So we don't have blinding, randomization, or placebo control. So the same group as its own control. We don't have a washout period. It means we don't have an interval between one dosage and the another dosage. So one can ask if we can have some cumulative effect here, and we don't know, maybe. And another limitation is that we are using healthy volunteers. Usually, it's better to evaluate antioxidant action when we have patients with inflammatory process or some other pathology that we can measure it better. Here it was healthy volunteers, and so no other pathologies and other concomitant drugs was used. So we are using healthy volunteers. That is a limitation of these studies. However, we have also the strengths that was the healthy young people, because it was possible to see in healthy young people that usually the oxidative processes are not so intense. We could observe the positive effect of propolis in this group, demonstrating that probably in the other most impacting cases, we can have better results yet. We have different dosages exposures. We tested 2 different dosages. We are using gold standards for detection of oxidative stress injury. That is a very positive impact in this work. And we are using a standardized propolis product. That is another positive aspect. So in the end, we can conclude that EPPAF has an important antioxidant effect in humans. This effect does not involve consumptions of endogenous antioxidant substances as vitamin E and glutathione system. There is a reduction of cell membrane damage and DNA damage. That is a very important result. And there is an increase in superoxide dismutase activity. To demonstrate an application of these important antioxidant effects in healthy volunteers, I found this publication that studied the cognitive improvement and safety assessment of a dietary supplement containing propolis extract in elderly Japanese. In this placebo-controlled, randomized, parallel group, double-blinded human clinical study, was possible to see the impact of the treatment of these patients during 24 weeks using propolis, that it was possible to have a better result in the memory and the verbal memory, demonstrating that antioxidant effects that we observed in healthy volunteers can be extrapolated in the case of degenerative diseases possessing important impact in verbal memory and processing speed. On the other hand, we have also animal studies. This study from our group we published recently, this year, demonstrated the impact of the use of propolis in Parkinson's disease, in this case, demonstrating the potential cardioprotective effect. This study concluded that these pioneering findings bring new insights into the effects of propolis on cardiac metabolism in experimental Parkinson's disease and suggest potential targets for further studies in this field. Treatments with green propolis as a dietary supplement is suggested as a potential candidate to minimize non-motor manifestations of Parkinson's disease. It's another thing that we can have in mind. We did also a clinical trial in the case of chronic kidney diseases. And in this study, we demonstrated the impact of propolis treatment for during 1 year in the reduction of the proteinuria, demonstrating the important effect of propolis in this. The conclusion and the discussion of this work talked about the possible involvement of antioxidant effect of propolis in this mechanism of action. Also, in this clinical trial that we published using EPPAF in COVID-19 patients, one of our conclusions involved the participation of antioxidant and anti-inflammatory effect of propolis EPPAF in the outcomes observed. I would like to share with you that this year we are celebrating 40 years of Apitherapy, and we are working with several partners in the universities in Brazil and outside of Brazil, as Portugal, Japan, France, and some other countries. And we we are present in more than 40 countries. We are now a member of Learning Group, and we are considered the leader in Brazil in the propolis extract field. I would like to express my special thanks to all my team that work hard to have everything that we are doing in the clinical studies and in vitro and in vivo studies in the development of our products. This year, also to celebrate these 40 years, we launched in the market the kids line using the iCaps technology. We are microencapsulating propolis in order to mask the taste, flavor, and smell, and improve the absorptivity, the bioavailability. And so this year we introduced this very beautiful and impacting line with great innovation. And also we introduced our line of products in Europe, especially in France and Portugal until now. In 2019, we did a celebration to share the results of our research team, using lectures and poster presentations to share the results of around 80 members of our research team, internal and external. And in this newspaper called Folha de São Paulo, that is a very impactful one, we was listed as an impacting company doing research with universities. We were in front of several big companies as Sanofi, Microsoft, demonstrating that what we are doing is very serious, is with very impacting quality, and we are demonstrating scientifically the benefits of propolis under several points of view. Our research are being supported not only by Apitherapy, but for FAPESP, CNPq, and FINEP, that are agencies that support research in Brazil. Thank you very much. It was a great pleasure to be here with you, sharing what we are doing. I would like to have the opportunity to be in a presential meeting as soon as possible, maybe in Australia. And I would like to invite all of you that can be in Brazil to visit us at Ribeirão Preto, São Paulo State. It will be a pleasure to share our laboratories and our facilities to you and show what we are doing. Thank you very much for everyone that is here watching this presentation, and especially for the organizer of this great event. Thank you very much.